So hi, I'm Eran. I'll tell you today about two studies that were done as part of my PhD at the lab of Uri Alon at the Weizmann Institute, where I had the privilege of playing in science for a few years. These two studies were done in tight collaboration with two colleagues and great friend of mine, Ariel Cohen and Naamage Vazatorsky. So we were motivated by the following question. If you take two cancer cells that have just divided from the same mother cell and you expose them to chemotherapy under the exact same conditions and monitor their behavior through time, what you will often see that one of the cells survives while the other one dies. And we asked why. Why these seemingly identical cells show such a different response to the same drug. Now, to begin and address this, we've developed a system that enabled us for the first time to watch the proteome dynamics of over 1,000 different proteins in space and in time in individual living human cancer cells. And one of the findings that we were able to identify is that in response to the drug, for most of the proteins, individual cells showed a qualitatively similar response. They went up together and they went down together in terms of their protein content. Diverging from this norm were a subset of proteins that showed a remarkable behavior. In the beginning, the population behaved in a uniform manner, and then, after some time, there was a bifurcation point. Some of the cells decreased their protein content, while others increased. And we were able to map that to the fate of the individual cell. After I completed my PhD, I co-founded a startup company called Mime Diagnostics, where we were developing disruptive technologies to try to assist physicians to reduce antibiotics misuse. And I hope that the networking that I've done during this wonderful time here at Stockholm is going to assist me later on in trying to take our work and to publish some of the scientific results that we were having. And also I hope that the credit that comes with this prize is going to help us open new doors and potentially new collaborations. Okay, so I started as a computer engineer, and at the end of my second year I went to do student exchange at DTU, the Danish Technical University, and on the way to the university there was this biological center called CBS, Center for Biological Sequencing. So one day I gathered my courage, I knocked on the door, went inside and told them that I would like to perhaps participate in a project. I didn't have any background in biology whatsoever. I just read one book by Richard Dawkins, The Selfish Gene. But they gave me the opportunity. I started at the beginning, I really didn't know what I was doing, but I think after half a year we came upon an interesting finding, and the head of the center, Sam Brunak, offered me to come during the vacations to fly to Denmark and to start doing to write a publication, and that was when I was first exposed to this fascinating process of doing science. So that's when I decided I want to pursue this avenue. So I think that in 2 or 3 years from now, if we could take the product that we're trying to develop right now and really bring it to the clinic and really impact the life of patients, that would be extremely gratifying for me.